Hi, this is Dr. Steven Feldman. I'm professor of dermatology, pathology, and public health sciences at Wake Forest School of Medicine in Winston-Salem, North Carolina. This is the Gray Line, in which we will discuss key characteristics of biologics and the two pathways for developing a biologic in the United States. We'll also learn that biosimilars are not generic versions of the reference biologic. There are major differences in how generic small molecule drugs and biosimilars are developed. What is a biologic? Biologics are viruses, therapeutic serums, toxins and antitoxins, and similar products that are used for the prevention, treatment, and cure of diseases in humans. They're derived from living sources, so typically they could be either derived from a human or animal or made in cultured cells, bacteria, or yeast, or other cells. And ultimately, from these come our therapeutic proteins. Biologics are large molecules, and they're very complex. Their structures are much larger and more complex than the small molecule drugs that we've traditionally given to patients. You can compare here aspirin versus insulin. Insulin is considerably more than 10 times as large. Growth hormone is another factor of 4 larger than that. And the monoclonal antibody drugs are absolutely enormous, maybe 1,000 times larger than an aspirin molecule. And they're complex structures. Typically, we think of them as protein molecules, but they're glycoproteins, so there's going to be sugar molecules attached to the chain that create additional potential for variability. And the folding of these complex molecules may represent another area where there can be complexity. Now, some variation is acceptable within and between medications. So generally, if a drug provides 80 to 125% of the blood level at a 90% confidence interval, that's considered acceptable similarity. Biologics provide a more complex question because it's not just the amount of the drug in the blood level that has to be within an acceptable range, but the variability in the complex molecule has to be similar as well. The biologic production process is complicated and has a lot of room for potential variation. To start off, we're going to try to make a protein, but we start with the gene for that protein. Now, that can be made identical. You know what the genetic coding sequence is, so if you were going to make a biologic or a biosimilar, you would use that same genetic coding sequence. You would insert that gene into some cell that would grow and make the protein. Now, there's a lot of potential variation in those host cells. And then those cells are cultured under very precise conditions, but tiny little changes in those conditions could affect the carbohydrate that gets put onto the protein, the tertiary structure, the folding of that protein molecule, the charge on that molecule, and whether that protein aggregates or not. This is followed by a purification process designed to remove the host cell DNA and other proteins, the aggregates. The protein is eventually concentrated and put into some final formulation, but again, there's a lot of potential for variability there. Ultimately, you get this final biologic that's a complex protein with sugar molecules on it and a variety of charges. The biologic product is so complicated that nobody can duplicate it, not even the originator company. And so these biologic products vary over time. Part of that variability will be due to changes in the source material that are used, if there was a change in the cell line or the purification procedure. There's going to be batch-to-batch variability. So one batch is not going to be identical to the next batch. The manufacturer needs to show that the product is comparable. It can't be identical because nobody can duplicate these structures completely perfectly. Batch-to-batch variability has been carefully assessed for different batches of the product etanercept. Here you can see a variety of studies, including differences in the percentage of basic variants, differences in the glycoprotein structure, and these vary from batch to batch, and even over time, patients might go, at one time might be on one batch and then switch to a different batch. Changes in the production process can lead to differences. In section C there, you see the change in the basic variants that happened after a change in the production process. I think when we think of biologics, you think this is a single drug. But, in fact, if you run it through a separation column, you find that it's a mixture of different structures with different glycoprotein structures. And then from batch to batch, those structures might change. Let's consider how biologics differ from small molecules. Well, first is the size. The biologics are enormous, and the small molecules are really quite small, certainly in comparison. The structure is very different. A small molecule has a simple, well-defined structure. A biologic has a complex structure that has folding and carbohydrates added to the protein structure. The protein sequence for biologics will be the same from batch to batch, but how that protein folds and what post-translational modifications are done to it can vary. Small molecules are relatively stable, whereas biologics, which are large protein molecules, are sensitive to the storage and handling conditions. The manufacturing process is different. Small molecules are synthesized by a predictable chemical process, and so you can make the same thing over and over again. As we saw with biologics, they're produced in living cells, and by a complex process, complex purification afterwards, you can't make identical copies from batch to batch with this approach. Immunogenicity can be different. With small molecules, we don't have to worry much about immunogenicity. With biologics, we do. And to characterize these, small molecules are relatively simple to fully characterize, whereas biologics would require a host of studies to fully characterize the molecule. Let's define some of what we're talking about with the small molecule drugs. When you make a generic of that, it's basically chemically identical to the branded drug. The blood levels that you achieve when giving those drugs have to be within a certain range, and the chemical is identical for all practical purposes. We're going to be focusing on biosimilars. So what is a biosimilar? It's basically a copy, as near as you can make one, of the commercially available reference product. A reference product that's no longer protected by patent. And these biosimilars undergo very rigorous analytical assessments to make sure they're similar to the reference product. And in addition, they undergo some clinical assessment to absolutely prove as best you can that it's going to perform basically like another batch of the reference product would. And they're approved by the regulatory agencies according to a specific pathway. So the biosimilar is highly similar to the reference product in its physical-chemical characteristics, its efficacy, and its safety. Now, biosimilars are not biobetters or second-generation biosimilars that are different from the original. These second generations are structurally different. They're intended to perform better, maybe using the same mechanism of action. So, for example, you had infliximab and then adalimumab. Both are TNF inhibitors, but they're very different drugs. They're not biosimilars. Biosimilars are also not generic drugs in the same way that small molecules differ from large molecules. Generics differ from biosimilars. So the small molecule generics are so much less complex that they're basically identical and they're regulated under a different pathway.
Now that we've learned about biologics and differences between generic small molecule drugs and biosimilars, let me summarize what we've learned. Biologics are so large and so complex that nobody can duplicate them, not even the innovator company. Biologics vary from batch to batch. Now, biologics have been revolutionary for our management of inflammatory diseases, and the batch-to-batch variation, not something that has affected our use and probably something we didn't even think about. Biosimilars are similar to the innovator biologic. We have piles of evidence showing the similarities between the biosimilar and the innovator, far more information than we have on the amount of variation between different batches of the innovator product. In some sense, I like to think of biosimilars as behaving as we might expect a different batch of an innovator product to behave.